So you've launched your comprehensive genome analysis job, and we've looked at the upper level files. So let's just go look and step where we've been. So I click on my jobs page. I want to go to an earlier job that I did. I could filter here by clicking on comprehensive genome analysis. And let's look at this job. So I highlight the row, and then I click on the view button in the vertical green bar to the right. In an earlier video, we discussed in detail the full genome report and what all of these downloadable files are that come with the comprehensive genome analysis. This one, when we submitted it, we used reads. So we have an individual assembly job. And we also have an individual annotation job. Now, earlier we talked about what you see when you click on the checkered flag here to go see the assembly job. In this video, we're going to talk about what you see when you click on the annotation job. So we click on it here, and it opens up, and there are a boatload of files here. If you've looked at any of the annotation jobs that are the videos that I've presented for just the straight up annotation service, I go into a lot of detail describing these different files and the formats that are there. I'll just do it briefly here. The contigs are here, the FASTA files, and you can download those. If I click on it, you see I can download it. There are a number of things I can do with that. This is an Embl format of annotation. This one has the feature DNA FASTA. So if you open that up, it's going to have each of the genes and the DNA sequence for them. This one is the same thing, but this time it's the protein sequence. This is just a text list of the features. This is the GenBank format. This is the genome format. This is another a GFF file. This is the merged GenBank format, and then you have it all wrapped up in a tar-GC. This one I sort of skipped over annotation genome. It just gives you, it's a JSON file that shows all the information associated with the annotation. The text file is a summary of that. You can get this Excel file, which I would encourage you to download and look at. It shows you all the information about each of the genes that are called and ends with the sequence. And then there's the quality JSON file. Now you notice I skipped over this one. This is the, all of the files are important, but this is a really important file within the annotation job because it tells you a lot of information about the quality of your genome. So I've highlighted it, and you can click the view button. And this is what it tells me. It tells me the genome ID that it has in Patrick, what the genome is, what the reference genome was when it's looking to see how good of quality this genome is. It wants to see this it's comparing it from this particular genome, and you could click on that, and it would open up a new tab and show me just exactly who that genome is. Sometimes we have to wait a little bit for Patrick to show up, but it's showing me this was the reference genome that it used. It says the coarse consistency is 98.7. The fine consistency is 98.4, and it gives me estimates of completeness and contamination. So I feel pretty good about this genome. Here's the evaluation group. It tells me how many contigs, and then a whole bunch of different parameters on estimating the quality of the genome, the N50, the L50, and this is the interesting one, where it's looking at the Staphylococcus group and that reference genome, and it's asking, does it have any genes that are unique to it, and is it missing some genes that it shouldn't have? So we have over-present, under-present roles. The roles that are predicted, the completeness roles, the total distinct roles, and in Patrick speak, roles is the genes. And then it tells you the genes without a functional assignment, meaning the genes that are not called hypothetical, and the genes with a functional assignment. How many of the gene feature coverage or protein encoding? How many are hypothetical? Actually, that's pretty good compared to most of them have actual function. And the features that are in the local protein families. I've talked exhaustively about global families and local families. Local families are those for a specific genus down. So the potentially problematic roles are Patrick has scripts that are running to look at your genome, and it's saying what do we expect and what don't we expect. And so it looks like this particular gene. It predicted that this genome would have 1, but it has 5, so it got flagged. 5 of this. Immediately it seems to be a problem. And then it tells you where those are and what the problem is. This is actually so valuable. Look at this comment that it has. Here's this gene, number 8. It has no good roles and is short, meaning that it's probably not a good gene. And right next to it, number 9, no good roles is in short. Number 10, no good roles and is short. This is an indication to me that these are all on the same contig, and there's probably either some assembly problem with this, or this is truly a gene that has issues and it's becoming pseudogenized. And it may be pseudogenized because there is a perfectly good gene somewhere in the genome. Like here, this one looks like it's pretty good. And here's another one that this is the one it's calling, and it's looking at that reference genome. Look at this number, 127.6, and if we go up here, the 127.6, it looked at those 2 genomes and it says, you know what? It's really close. It's got 869 out of 1,000 matching k-mers. We think that this is the one that did it, and the rest of these ones are probably no good and junk. You could dig into that if you wanted to by clicking here, and that'll take you to the page for that particular feature. It's going to the genome browser or the compare region view. I'd be more interested in looking at the genome browser when it comes up. But this is the overview for that gene. And the genome browser should show me all those little pieces when it pops up of those individuals' genes. So it's just taking its time to load on that. But that just shows you how you can take from this. And this is how valuable this report is. It's showing you all this information and what you should expect. I think by far it's one of the best reports that we have in Patrick that really allows you to dig in what's going on with your particular, the genome that you used in this service. Now, in our subsequent videos, we're going to talk about how you view this genome in Patrick. So you have the report and everything, but what you really want to do is see your genome in the context of Patrick and what you can do with it once you get there. So we're going to show you that. And then we'll also show you where you can go when you need help. So see you next time.